My name is Charles Runckel. I am a grad student in the DeRisi lab. I'm entering my 4th year of 6 or 7 years of graduate school now. I work on the honeybee colony collapse disorder. In the last 3 or 4 years, a large number of honeybee colonies, farmed honeybee colonies, have been dying. We use honeybees to pollinate a large amount of our crops. Pretty much most fruits and nuts are pollinated by honeybees. What was happening is that starting about 3 years ago, 70 or 80% of the hives would die, and you start having farmers not being able to pollinate their crops. And our main project is taking a commercial beekeeper who has 72,000 hives that he ships across the western United States, to take 20 of those hives and to sample them every week for over a year, and then to run our pathogen diagnostic on those hives. As a part of this project, I've taken a number of trips to go collecting bees. And so I had the full bee suit, big white suit with the mesh, and we went out there. We've had some very great collaborations with beekeepers. They're very passionate about the welfare of their bees, and they're very interested in what we do. We actually train their people to do this, so as they travel around the country, they collect the samples and send them to us. Honeybees have bacteria in them naturally. They may have fungus. They may have a virus infection that they just picked up because they sampled some pollen that had a virus on it. Then the question is, how is it getting all these different viruses? What vectors are there? What other parasites are there? And just figuring out how much a colony can withstand if it's some combination of these factors that's causing the colony to collapse. The genesis of this project was that there have been a number of high-profile papers that said it's this or it's that, and the science wasn't definitive. We don't know what the root cause of colony collapse disorder is, and the point of this study is to figure out, well, what's supposed to be there normally? What's supposed to be there normally, and to expand the list of pathogens we know about, because it's very hard to discover a new pathogen. That's why we have all these microarrays and deep sequencing. But once you've discovered the pathogen and you have its sequence, well, then you can share that with everyone, and you can design very easy, simple PCR assays, and then everyone can look for this and determine what's causing what. And so we designed a new bee pathogen chip to look at bees. So we have some idea of what's normal in the background of a bee, what's exceptional, what things seem to pop up together. It's not just a bee virus chip, it's a bee pathogen chip. So it includes all of the known fungal pathogens of bees. It includes all the known bacterial pathogens of bees. It includes all the microscopic mites and ticks that a bee can get. It includes various types of flies and wasps that can lay their eggs in bees and kill them that way. So it's a comprehensive pathogen chip. I get bees generally that are shipped to me from the bee farmers. I extract the nucleic acid, I hybridize it, and then we analyze that microarray data. And then following the microarray data, I confirm interesting results with PCR, and we're gearing up now to do ultra-high throughput deep sequencing on the bees and get tens of millions of sequences of hopefully novel bee viruses. People have been studying bee viruses for about 40 years because bees are so important to agriculture. The beauty of using the Virochip is that it detects not just the virus that's been studied and sequenced in GenBank, it can detect related viruses. So it's not so important that we have the exact virus on the chip, just that we have a virus in the same family. So we can, for example, look for a virus that has never been discovered before in bees, but whose relative has been discovered in the fruit fly. We use things like quantitative real-time PCR or deep sequencing, and we can say, well, not just that this bee has this virus, but how much of it has. And so if it has an extremely tiny amount, well, we can say that virus is there, but the bee probably isn't really infected. And if it is there in a large quantity, well, we can say there's no way that rubbed off. It must be replicating. One of the most promising hypotheses is what I call Nosema plus. So Nosema is a microsporidian. It's a very basic fungus. The pathogen in question is called Nosema cerana, and this is actually a jump over from the Asian honeybee. In America, we use the European honeybee. And this fungus is very benign in the Asian honeybee, but when it crossed over to the European honeybee, it became highly pathogenic, but not pathogenic enough to explain all the colony collapse. But what seems to be happening is that the bees are getting Nosema cerana, and then they're getting other pathogens at the same time, and perhaps some combination of those are what's causing the collapse. We have been tracking another parasite called Crithidia mellificae. It's a distant relative of malaria, so in many ways this is honeybee malaria. We have been observing dozens of instances where the honeybees are infected with Nosema and Crithidia, or Crithidia and viruses. And the Crithidia seems very much associated with the infections of Nosema and with the viruses. So it may well be causing or be caused by one of these other infections, or it may just indicate some type of disease state of the bee. So there are more unicellular pathogens to the bees than people have been looking at so far. And so that's been an interesting case. The really interesting one has been a collaboration we had with San Francisco State University, and that's been phorid flies. Phorid flies are a type of parasitic fly that affect many different organisms, one of the most famous ones being ants. Phorid flies are sometimes called zombie flies because in ants, they inject their larva into the thorax or chest region of the ant. And within a couple weeks, the larva crawls into the ant's brain and eats the ant's brain. At which point, what's left of the ant's nervous system just makes it wander aimlessly, like a zombie. About 2 weeks after that, the phorid releases enzymes that digest the ant's neck, causing the head to fall off, which is also why they're known as decapitator flies. Now, this was known in ants for decades, but our collaborators at SFSU found the first phorid in a honeybee. And so now we're looking back into our historical databases of bees and looking to see, well, are the phorids contributing to colony collapse? Are they weakening the hives? So that's the big thing we're following up right now, both in terms of the phorid as a pathogen itself and the phorid as a vector for other bee viruses, or for unicellular pathogens of bees.